it's my great pleasure to introduce our second speaker for today. Hlib Razumkov was born and raised in Kiev. He studied at the University of Toronto and University of Tokyo, where he graduated with a Bachelor of Science degree majoring in chemistry, and joined the Stanford Chemistry Department in 2020. He pursues his PhD studies with Nathaniel Gray Lab, focusing on designs of the modular scaffolds for efficient synthesis of proximity-induced small molecules and molecular glue degraders. Thank you very much for joining us today, and the floor is yours.
All right. Thank you very much for this introduction and for inviting me. And today, I will be talking about discovery of CRBN-dependent WEE1 molecular glue degraders from a multi-component combinatorial library. So given the previous speaker, I believe most of my intro would be glanced over, but essentially we know that the traditional drug discovery very much focused on development of individual protein ligands and using them as inhibitors, hence being successful for some diseases, but having a very limited percentage of overall disease relevant protein targets that are left accessible to this modality. And so additionally, it also relied on stoichiometric inhibition, which requires higher amounts of the active agent and may lead to potential off targets. In the event-driven pharmacology, a more recent approach in drug discovery, which was just presented as well, the focus is more on recruiting a protein which already exerts a particular function within the cellular machinery and sort of rerouting that function towards the protein that we would like to affect for a therapeutic benefit. However, obviously, the question remains, how can we accomplish that without relying on the magical AI-generated protein transporting scientists? And one of the answers was provided previously in the field of targeted protein degradation, originally when the proteolysis targeting chimeras or PROTACs bivalent degraders were reported, where an E3 ligase protein would be brought in close proximity to the protein of interest by a bivalent molecule which has specific and relatively potent ligands for either of the targets connected by flexible or rigid linker, and then leading to ubiquitination of the target and its degradation. And while this is indeed eliminates all of the activity of the protein due to its degradation, perhaps except for some degraded peptide activity or its MHC presentation, it also allows us to rationally select which protein we would like to degrade. And unfortunately, there lies the cons of this approach, that we really need a ligand, a specific ligand for that protein of interest, which limits the scope of proteins that we can degrade, and also the bivalent compounds have a little bit of stability and cellular permeability, though this has been combated and greatly improved over the last several years. So for the molecular glue degraders, in particularly focusing on the cereblon degraders, the compounds would not bind either of the pockets and would not really rely on the pocket, but rather bind a pocket generated by the two proteins. And in this way, a neo-substrate, so a non-canonical substrate of an E3 ligase can be recruited and degraded. And of course, the pros of that are that we don't need the exact ligands with nanomolar potency for either side, and they have better pharmacokinetic properties and more drug-like than bivalent degraders. But again, the discovery remains serendipity-driven majorly. However, as we've just seen, this begins to change. And here I present several different compounds which were previously reported, and they pretty much all had glutaramide moiety, which has just been slightly debunked as the only cereblon engaging sort of moiety necessary for recruitment. And however, the fact is that we can definitely elaborate and explore the chemical space outside of the glutaramide or glutaramide-like recruiters, leading to a wide range of cerebral neosubstrates reported, over 100 in the papers which focus on the scale of those neosubstrates, as well as several, like under a dozen, which are the primary targets present in current clinical trials or close to clinical trials, therapeutic agents. However, the majority of reported derivatives are usually obtained through a very scarce and limited synthetic perturbations. Hence, the synthetic routes may make the diversification of these compounds slightly difficult. And especially since majority of the older compounds have been reported in the synthetic pathways, which would lead to linear and rather flexible molecules. So in my effort, which may or may not be beneficial, but essentially in my effort was to find more facile way to expand the cerebellum degradome and yield potent and hopefully selective molecular glue degraders. And in order to do that, I've focused on the multi-component reactions in the molecular glue library design. And here I've generated a library of over 200 compounds relying on this MCR approach because it provides easily diversifiable core, which is usually substituted in 3 or 4 different places. And those substituents can be modified somewhat readily. And hence gives access to a more 3-dimensional chemical space around the scaffold. However, today I will focus on the scaffold derived from one multi-component reaction. In this case, it is the Grobke-Blackburn-Ganmei or GBB reaction presented over here, where an isocyanate, aromatic amidine, and an aldehyde are mixed together to generate this imidazopyridine core, which interestingly enough was also featured in the previous talk. So maybe there's something about that. And I added the glutaramide derivatives onto the aldehyde portion of the molecule by introducing these precursors and coupling them with a range of commercially available amidines and isocyanates, leading to resulting library. And in this case, the compounds which are highlighted here are the precursors which were used in the short version of medicinal campaign that I'm about to present here. So we've used the simple proliferation screen using the CTG assay, cell titer glow assay, and we've identified several compounds which were highly cytotoxic, up to 5 nanomolar EC50 in the MOLD4 cells, which is the blood cancer cell line of choice. However, their cytotoxicity was completely rescued in the cerebral knockout version of the cell line, and the GSPT1 degrader here is shown for comparison. And that made us curious about the culprits of the effect that we observed. So we picked the most cytotoxic compound, compound 2, for the subsequent proteomics. And thankfully, Catherine, the God Almighty of proteomics for our lab, was able to generate the following plot for us. And we've seen that WEE1 and CK1 alpha were the primary targets of the molecule shown over here after 5-hour incubation at 1 micromolar. And additionally, this was validated by Western blot, conducted by Zhixian, another very significant contributor to this to the biology of in this project. She was able to show that all three compounds degrade all three proteins, albeit to a different extent, and all of that degradation was completely rescued in the cerebral knockout cell lines. And we've also been able to show rescue with the ubiquitination, clean ligase dependent ubiquitination inhibitor, MLN 4924 over here, which in this case is a partial rescue, but as we repeated that plot separately with our hits, like modified hits, we see complete rescue at higher concentrations of the MLN compound. And so here I'd like to take a quick step back and just talk about the WEE1, the new target, since CK1 alpha has been previously reported and was degraded by both Christina Wu's compound, lenalidomide, and as well as the efforts in St. Jude, which were published earlier this year or late last year. So the role of WEE1 is primarily in the G2M transition. It is a checkpoint kinase. When protein, when the DNA replication leads to DNA damage accumulation, the ATR kinase senses that, and then through a phosphorylating cascade activates WEE1, and through phosphorylation inhibits the inhibitor of WEE1, CDC25, leading to subsequent inhibition of CDK1 activity by phosphorylating at the tyrosine 15 and threonine 14 residues, and stalling, pardon me, and stalling the entry into the mitosis so that the cellular machinery would have enough time to hopefully repair the DNA damage observed. In some malignancies, in particular lung cancer, small cell lung cancer, other cell cycle checkpoints are heavily mutated or truncated or at all absent, and that leads to bad control of replication and high genomic instability, high amount of DNA damage, which makes WEE1 inhibitors and degraders promising therapeutic approach, since inhibition of WEE1's activity under these circumstances would lead to mitotic catastrophe, which would hopefully be much more prevalent in malignancies which rely on that as its last resort. So there have been previous reports of the very least 3 WEE1 inhibitors, as well as a PROTAC developed by our lab from the AZD compound over here. And they've all shown both WEE1 activity downregulation as well as negative effect on the cancer cell proliferation. And despite that, it still remains important to continue development of the molecular glue degrader for this target, which may prove useful to be a supplementary strategy, if not primary strategy, if the active site mutants are observed. Since as I'll show you later, the molecular glues that we've identified do not engage do not engage WEE1 through the active site. So we utilized the hybrid system and generated hybrid tagged cell lines for CK1 alpha and WEE1. And then we use that for screening to identify if we can generate selective WEE1 degraders. So quantification of our original hits shows single-digit nanomolar DC50 values, suggesting that it's a highly potent degrader for both targets, and the methylation on the glutaramide, which is known to abrogate the activity, indeed does so. In the interim, compound 2 has shown, as expected, a slight preference for CK1 alpha degradation over WEE1. And you may see some discrepancy with the Western blot, since or proteomics, since WEE1 was downregulated to a lower extent, it is important to keep in mind that these are the Jurkat cells rather than the MOLD4, and we've seen such slight discrepancies, which usually are in numbers, but not in quantitative, but not really qualitative aspects, since indeed the preference for CK1 alpha degradation retains here in both DC50 and Dmax. And the third compound, which demonstrate both weaker degradation potency for both targets and also demonstrate slight downregulation of GSPT1, the bane of existence of every cerebral molecular glue developer, sort of made this scaffold less attractive for us. So we focused on this amino imidazopyridine scaffold in compound 1 and 2 for further development. Based on those results, I first changed the isoindolinone linker to a phthalimide linker, and we've seen a general partial loss of potency. However, slight preference towards the CK1 alpha, WEE1 degradation over CK1 alpha, and substitution change of imidazopyridine residue from position 5 to position 6 over here led to complete rescue, showing that the exit vector here is, of course, very important. And interestingly, Chishan, who helped me, who's a postdoc in lab, who also helped me with synthesis of some of the derivatives in our pursuit for selective WEE1 degrader, was hoping to generate an amide bond between this carboxylic acid and this amine. And interestingly enough, we were surprised that the compound that was generated was not only the amide, but also a selective WEE1 degrader over here. So that sort of makes it slightly funny, because we are hoping for a more rational discovery, but I guess serendipity finds its way no matter what you do, every so often. And so going back to compound 1 and 2, while the studies from the previous slide were ongoing, I wanted to further scrutinize the amine substituents, and changing it to a chloride or leaving the bare amine with unsubstituted amine did not significantly impact the DC50 values for either of the protein. It did not lead to any selectivity, suggesting that it is truly the substituent that further increases the potency. And it was indeed the case, because when I introduced rather bulkier substituents, 2-dimensional or 3-dimensional in the case of this adamantyl, we've obtained some of the strongest degraders, and in the case of compound 10, the strongest WEE1 degrader in our list, which I'm presenting today, both by DC50 and the Dmax. And then I decided, well, why not take it to a further extreme and further elaborate on this hydrophobic residue. And additional extension here actually led to some WEE1 preference, or rather loss of CK1 alpha degradation activity. And remember, the compound 4 was the phthalimide moiety also had a similar effect. I've synergized them, and in the combined compound, we were able to see. We were able to see even further selectivity improvement over here, and addition of a further steric bulk in that region leads to complete selectivity over CK1 alpha, albeit at the cost of potency. So then we turned back to Catherine, and she helped us run proteomics on these two hopefully selective and actually indeed selective WEE1 degraders. As of now, we pretty much see WEE1 as the primary target, with other less relevant proteins observed over here. And thankfully that confirmed a large extent of selectivity that we see not only over CK1 alpha, but over the proteome as well. And the next step was taken by Zhixian, and she wanted to observe whether or not we can see the cytotoxic effect or antiproliferative effect on a range of leukemia, lymphoma, and colorectal cancer cell lines. And while we've seen some, both by the control PROTAC and GSPT1, as well as the non-selective degraders like compound 10 here, the selective degraders unfortunately did not have an EC50 below 10 micromolar, and their treatment at 10 micromolar resulted in just modest downregulation of the cellular proliferation. And then we thought to ourselves, maybe we have a low, the essentially sacrifices we had to make in potency in favor of selectivity made this degrader not potent enough for to induce the cytotoxic effect, and probably the levels of WEE1 which remain are sufficient for the cellular survival. And we wanted to see if we could increase the levels of the DNA damage that might may or may not improve the antiproliferative effect of these compounds. So we tried to co-treat the compounds, the selective compounds, with gemcitabine, which is a chemotherapy DNA damaging agent. And we've actually seen slight synergy with compound 6, which was the stronger of the two degraders in MOLD4 cells and in Jurkat cells. And this effect is rather minor in MOLD4T cell line. And the hypothesis that indeed we may not be degrading WEE1 sufficiently sort of tracks with the PROTAC, the similar experiment with the PROTAC co-treatments, where the synergy which was greater, likely due to the higher potency of the bivalent degrader. And additionally, we wanted, of course, to rationalize how WEE1 is being degraded and what is the G loop or beta hairpin with a glycine that we are recruiting here. And just by looking at the WEE1 kinase domain, there was a very clear candidate for us, which is both sterically homologous with CK1 alpha's G loop, this glycine 322. So insulin Zhixian mutated it to alanine and asparagine residues, so site-specific mutagenesis, and resulted in complete rescue or partial rescue with the alanine, probably suggesting that small steric bulk here could still be tolerated, probably due to a larger hydrophobic contact that we'll see in a second. So we decided to further elucidate the WEE1 recruitment mode by obtaining a structure of its ternary complex with cereblon. Zhixian ran the TR-FRET assay on the labeled WEE1 kinase domain that we had, and the cereblon DDB1 that was kindly provided by the Fisher lab. And the screen revealed that compound 1 and 10, which are both strong WEE1 degraders, are indeed not showing the hook effect, and compound 10 shows the highest TR-FRET ratio here, while we see a clear hook effect for the PROTAC, albeit at a lower TR-FRET ratio. And that helped us select our candidate for the structural studies. And then, thanks to the diligent cryo-EM experiments performed by Key in the Fisher lab, we were able to obtain the ternary complex structure shown over here. The engagement of WEE1 and DDB1 was rather flexible, and cereblon existed actually in both open and closed conformations, as observed by the cryo-electron microscopy. However, despite these flexibility, Key was able to identify two potential compound binding modes, shown over here, with adamantyl either pointing towards this isoleucine or the end of the imidazopyridine. But in both cases, we present a hydrophobic residue towards this isoleucine, which is in its own way hydrophobic, while the compounds, which are CK1 alpha degraders, lenalidomide, or recently reported as J3149, while having a very similar presentation of the lenalidomide's parts, the isoindolenone, do not necessarily extend in that direction. And also the CK1 alpha has glutamate in the sterically homologous spot of that isoleucine in WEE1, which further rationalizes at least the compound 13 selectivity, likely since if this is the binding mode, further extension of hydrophobic bulk would interact with the negative charge counterproductively. So to conclude, we've synthesized, we used the multi-component reactions to synthesize a diverse library of molecular glue candidates. And with that, we were able to expand the cereblon degradable proteome by identifying the first reported in the published and peer-reviewed literature WEE1 degrader, molecular glue degrader, pardon. And we were able to generate some selective compounds with, which degrade WEE1 selectively. And the current efforts in the lab continue in the direction of rationalization of recruitment of WEE1 and usage of that to deepen our understanding of how we can recruit other kinases to cereblon and successfully degrade them. We hear the reports from industry about them all the time, and how we could hopefully devise a way to change the selectivity to our advantage. And we hope to further expand the number of kinases that we can degrade, as I said before, and we're currently continuing to screen this library in other non-cell killing based assays. So with that, I'd like to close and thank the Gray Lab, my PI, Zixien, who was an absolute trooper continuing the biology on the project after Inchul, who started a transition, Jishan, who helped with SAR, and the Fisher Lab, and in particular, Catherine, and especially Key, who did tremendous work in the structural studies. Thank you.